Okay, so let's start with the experiment. Yes. Now see, the title of the experiment is to study anti-ulcer activity of a drug by NSAIDs induced rat model. Okay. So basically what we are studying, this is, first of all, this is a preclinical experiment. Okay. Why preclinical? Because we are using rat. Okay, this is first point. Second point is in preclinical, we are going to observe anti-ulcer activity. Okay, anti-ulcer activity. So why we are finding out anti-ulcer activity even though so many well-established drugs are available? Okay, why we are going for anti-ulcer activity even though established drugs are available? Okay, established drug means they are well accepted for anti-ulcer activity. So the answer for this question is even though the drugs are available, but there might be a possibility. Okay, there is a need to go for the safe drug, safe as well as efficient drug. Okay, so this is what our requirement. The need is there is a requirement of new drug because of the side effects of the existing drug, and we have to search for the more efficient drug. So that is the need. So the third point in the title is we are assessing the effect of a drug. Okay. So this drug is basically a new drug. This is a new drug. Now this new drug may be synthetic drug, okay, or it may be herbal drug. Okay. So you can consider that this synthetic drug is synthesized in chemistry lab and this is extracted in pharmacognosy lab. Okay, so these two can be assessed with the help of this experiment. Now, to study the anti-ulcer activity, we should first induce ulcer into animal. Okay, ulcer. Now, this ulcer can be induced by NSAIDs. Okay, in last experiment, if you can recall, so we have used pyloric ligation method. Okay, we have ligated pylorus, and that was the pylorus ligation animal model. Okay, but in this we are using NSAIDs. Okay, and NSAIDs, as you all know, they are inflammatory drugs, anti-inflammatory drugs. Okay, so the main use of these drugs are to reduce the inflammation. Okay, but as a side effect, one of the side effect is they are causing ulcer. So that's why we are using the side effect in preclinical model. Okay, we are using the side effect of the NSAIDs to induce ulcer into animals. Okay, so how many points we have discussed? The first point is basically this is a preclinical model. First point. Second point is why we are assessing the new drug, even though the established drugs are present. Okay. Third, we have studied new drug means what? New drug means it may be synthetic drug, it may be herbal drug. Okay. Then, if you want to observe the anti-ulcer activity of the drug, so you should induce ulcer into animal. So that ulcer can be induced by pyloric ligation method or by NSAIDs. So as you all know that NSAIDs is a anti-inflammatory drug, but one of the major side effect is it is causing ulcer. So that's why we are using this side effect to induce ulceration. Okay, to induce ulceration. Okay, so this is a brief introduction of the title. Any question on this? Anyone? Any doubt? Yes, it is open for discussion now.
Sir, if we induce NSAID in other models, means other animals, then also the ulcer will induce.
See, See, we are using NSAIDs to induce ulcer. Okay, so you can use pyloric ligation also. It depends on you. So in previous experiment, we have studied this one. Okay, in today's experiment, we are studying this model. Now it's up to you. You can use both in different animals, different set of animals. Or you can use any one. It depends on you. If you're using both of them, so your results will be authenticated then. You can validate the result in both the model.
Okay.
Okay. Yes, any other person? Okay. Then we can move ahead. Now see, as we discussed, this is a preclinical animal model. So there has to be animal. And in this case, the animal is Wister rat. And the weight is 150 to 170 gram. Now you should
Sir,
consider.
slides.
Is it okay now?
Yes, sir.
Okay. Now see, the weight of the animal is 150 to 170. So you should take care of this weight. Okay, because we use this weight to calculate dose. Okay, so the weight is important. Now see, in case of drugs, omeprazole, indomethacin. So omeprazole is a standard drug. Okay, this is a standard drug, well-established drug. Okay, to treat the ulcer. Basically, it's an antacid. Then indomethacin. This is ulcer inducer. Ulcer inducer. Then we are using saline solution and 0.1 normal NaOH. Okay. So now see, the dose is 20 milligram per kg and 100 milligram per kg. Okay, so you can calculate for 150 milligram, 150 gram. Okay, just by simple cross multiplication. This is for omeprazole. So 150 gram animal. Okay, so you can cross multiply and you can find out the dose for this 150 milligram, 150 gram weight of animal. Okay, then we have equipment like Caesar and all. So one by one, we will try to see this use of equipment also. Okay, now I'm moving to the principle. See, in case of principle, NSAIDs, the examples are various examples are available like indomethacin, aspirin, ibuprofen. Okay, so these examples are there. So anyone you can take to induce the ulcer. Okay, only the condition is it has to be used in proper excess dose. Okay, excess dose. So if they are in excess dose, then only they will induce ulcer in animals. Okay, so the dose of this indomethacin, it is 100 milligram. 100 milligram per kg. So this dose will induce ulcer. Okay, this is too much. This is excess. Okay, अगर इसी को अगर आपने कम कर दिया जैसे 25 मिलीग्राम ले लिया तो this will not cause ulceration. Okay, so we are using 100 milligram per kg to induce ulceration. Right, so you can take anyone, indomethacin, aspirin, ibuprofen. Okay, all can be used. Now this is an important animal model in investigating potential usefulness of the anti-secretory and cytoprotective agent. Okay, so all these are the agents. Okay, agents, but they have not been assessed. So we are assessing these with the help of animal model. Okay, for example, if you have XYZ extract with you and you hypothesize that okay, this may be cytoprotective or this may be anti-secretory. So you can assess these two abilities, these two qualities of the drug with the help of animal model, this animal model. The underlying pathophysiology involves gastric acid secretion and mucosal prostaglandin synthesis. Now see, the underlying pathophysiology of this model is it disturb the acid secretion and it is disturb the prostaglandin synthesis. Who? NSAIDs. Okay, NSAIDs, they are disturbing the gastric acid secretion and prostaglandin synthesis That's why they are causing ulceration. Okay, so we will see in detail prostaglandin and gastric acid secretion. So they are most commonly used animal model for anti-ulcer study. Yes, so this is the first part of the principle. Yes, any question on this? Okay, if no questions, then we can go ahead. Yes, now the principle, second part of the principle is NSAID is reducing ulcer. As I told you, this is one of the problem, one of the side effects, okay, one of the undesirable effect of the NSAIDs. Okay, so if we are if we are studying the causes of the ulcer, causes of the peptic ulcer, so one of the cause is NSAIDs, okay, and another cause is H. pylori bacteria. So these are the two major causes of the NSAID, peptic ulcer. Now, NSAIDs are known to induce ulcer by inhibiting prostaglandin synthetase. Okay, prostaglandin synthetase. This is an enzyme which is responsible for synthesis of the prostaglandin. Okay. Now, PGs, that is prostaglandins, are found in many parts, like stomach also, and they are having protective role. Okay, by stimulating secretion of bicarbonate and mucus. Now you can see, for example, if this is a stomach. Okay, so PGs are creating a protective role, protective covering, okay, by secreting mucus. So this is a protective covering, or it is nothing but mucus. So this mucus is secreted by prostaglandin. Okay, so if prostaglandin is not present, so this will not be present, and then the protection will not be present, and there may be chances that there may be formation of ulceration. Okay, because the acid is going to secrete at normal rate, and if protection is not available, then it can cause ulceration. Okay, so this is one of the role of PG, that is prostaglandin. Then second role is it is secreting bicarbonate. Okay, now as you can guess that this is alkali. Okay, so this can neutralize the acid. This can neutralize the acid. So PG is secreting mucus to provide protection, and at the same time it is secreting bicarbonate to it help, and the bicarbonate helps to neutralize the acid. Okay. Now again, PG is important to maintain the mucosal blood flow and regulating mucosal cell turnover and repair. Okay. Now see, it is controlling blood flow to mucosa. Okay. Now you can imagine if this is not present, so blood flow will not be proper. And if blood flow is not proper, then mucosal cell may die. Okay. So to link this with this one, you can link this also. It regulates mucosal cell turnover. Okay, means synthesis formation of the mucosal cell. And again, it is helping in repair. Okay, so in short, PG is responsible for mucus formation, and mucus formation is responsible for protection. Okay, if it is not present, PG is not present, mucus will not be secreted and protection will not be present. Now this PG is inhibited by NSAID. Okay. So what we have studied in this slide, we have studied two causes of the peptic ulcer. Then second one, we have discussed the role of prostaglandin. Okay, role of prostaglandin. And And the role is, one is protection and the second one is synthesis of bicarbonate. Okay, protection means it is taking care of mucus cell. Okay, and what is NSAIDs doing? NSAIDs is causing decrease in Pg. So this will cause decrease in protection, decrease in bicarbonate, and this can cause ulceration. I hope it is clear. Yes. Now it is open for discussion. Yes, anyone? Is roll number 24.
Yes, sir.
Yes, sir.
Is it clear to you?
Yes, sir.
Now come to the procedure now. Okay. So in case of procedure, first of all, we are using female blister rats from 50 to 170, and then we are starving it for 48 hours. Okay. At the same time, we are allowing it to drink water ad libitum. Ad libitum means there is no limitation. Okay, no limitation. Okay, we are not restricting it to take water. So if they are starved, so they can take more amount of water. Okay, so ad libitum is means it is we are not restricting it. We are not, there is no limitation to take water. Starvation. Okay, why we are starving it? Because we want to avoid interference of food. Okay, then the animal groupings are like, first group is normal control group. So we will give saline solution for 10 days. Okay, PO, that is orally. Then ulcer control group, we will use indomethacin 100 milligram per kg single dose. Okay, only one dose will be sufficient to induce ulceration. Then, so in this normal control, there will be no ulceration. Okay, then in indomethacin, there will be ample of ulceration. Number of ulcerations will be there. Now, in case of standard group, we are using omeprazole 20 milligram per kg 10 days. Then at 11th day, we will give a single dose of indomethacin. Okay, so there has to be decrease in ulceration. Okay, because we are using omeprazole. In this case, we are not using any drug. We are using only ulcer inducer. But in this case, we are using ulcer inducer as well as the drug, anti-ulcer drug. So there has to be reduction in the ulcerative index. And the number of group is 10 animals will be there in one group. Okay, is it clear?
Yes, sir.
See, this is a diagrammatic representation. I have taken it from this research paper. Okay, now see, this is your indomethacin, and this is the structure of indomethacin. Okay, now we are inducing ulceration. We are giving this drug to the rat, and this is your ulcer control group. Okay, we are giving indomethacin, and then you can see there are a lot of ulcers, gastric ulcers are generated now because of this indomethacin. Now, we are taking this fruit, Cucumis melo, and then we are macerating it. Okay, maceration is a process to extract the drug. Okay, now this is a maceration, macerated liquid. Now, this is aqueous extract. So this aqueous extract we are giving to this animal along with this indomethacin. Okay. Now there has to be reduction in peptic ulceration. So this is your standard drug group, standard group, or this may be test group. Test group. Okay. See why I am differentiating it. Standard group means I have to use well-established drug like omeprazole. Okay. Test group means I have to use my own drug, like it may be extract or it may be any phytochemical, right? So that's why based on the drug, I will categorize it as a standard group or as a test group. Okay, but the procedure will be same. I have to induce ulcer by indomethacin and then I have to give the drug. So this drug may be omeprazole or this drug may be any extract. Okay, so one group is ulcerative control and another group may be standard group or test group. Yes. Any confusion on this slide? Yes. Any confusion on this slide? Any question?
No, sir.
Now see, this is the observation. So what you have to do is after indomethacin dose, after 4 hours, you have to sacrifice the animal. So you can sacrifice the animal with the help of carbon dioxide gas. Okay. Then you have to sacrifice the animal. Then you have to you have to take out the stomach, and then you have to clean the stomach, and then you have to pin it on a cork wood. And then ulcers will be scored using dissecting microscope. See, this is your dissecting microscope. Okay. And based on the grading, now the grading you have to score it. Now see, the grading is what? 0, almost normal mucosa, 1, vascular congestion, 2, 1 or 2 lesion, 3, severe lesion, 4, very severe lesion. Now, to minimize the error, okay, we have to go for a blind observer. Okay, a person who is expert in scoring, but he is blind. Okay, why we are calling him blind? Because he don't know about the groupings of the animal. You are handing over him sample and he is not aware of that sample, from which group this sample is given. So that will reduce the error. Okay, so we are using blind observer. Then in this case, what you have to do is you have to measure the mucosal damage area of the mucosal damage, and then total area. Okay, total area of the glandular stomach. Then I'll show you this one. See, this is control group. This is your standard group. This is your SEFS 15 milligram, 15 milligram. And this is your SEFS 30 milligram. Okay, so this is your test group, and this is also test group. Okay, now see, this is, I can consider this as a total area. Okay, and now this is your ulcerative area. So this area may be 50 and this area may be 10. Then multiplied by 100. Okay, so this will come around 20%. Okay, so this is your ulcerative index. Okay, now see, now come to this area, 15 milligram dose. Now this is your complete area. And now this is ulcerative area, this area, then this area, okay, this area. You can collectively, you can combine it, and then it may be like 5 divided by 50 multiplied by So this may be 10%. Now you can see in control, this is 20%, but the treatment is reducing it to 10%. Now this is again test group, but the dose is now increased from 15 to 30. So this percentage may be 5%. Okay, and this is your standard group. So see, okay, hardly you can observe any ulceration, but in this case you can observe this ulceration. Okay, so it may be 2% or 1%, 1 to 2%. It may be, I'm using the word maybe because you have to observe it carefully, and I have taken it from research paper. So you have to go through the research paper and see what is the exact figure for ulcerative index for this standard group. But logically speaking, highest will be there in ulcer control group. Then this has to be reduced in test group. Okay, so if we are comparing two test group, 15 milligram and 30 milligrams, so there may be highest reduction in 30 milligram group as compared to 15 milligram group. And in case of standard drug, this may have a maximum protection. Okay, so the percent of ulcerative area will be less. Yes, it is open for questions now. Yes, rule number 31. Is it clear to you?
Yes, sir.
Okay. Now see, this is your ulcer index. Okay, ulcer index. So I have given you direct example. I have taken 10 divided by 50 and then multiply by 100. Okay, so you can calculate any ulcer index. Then percent ulcer inhibition. So this is what? This is ulcer index in control, ulcer index in test divided by ulcer index in control. So control means you are taking 10 animal in one group. So you are calculating ulcerative index for 10 animal. And then you are taking average of this. Okay, and this average you are putting here. Similarly for test also, you are taking 10 animal, then you are calculating ulcerative index of 10 animal, and then you are calculating average. Okay? So why we are going for average? The answer is we want to reduce the errors. Okay, and we can get an exact value. So that's why we are taking average. So this is percent ulcer inhibition. Now see, this is one table. So this is for this group, ulcer control group. Okay, ulcer control group, body weight of one animal, it may be 150. Okay, it may be 165. Second animal may be 165. Third animal may be 175. Okay, so like that, the dose of indomethacin will vary. Indomethacin. Okay, because we are using this control, ulcer control group. In ulcer control group, we are giving only indomethacin. So the dose of indomethacin will change. Then at the end of 11th day, we will just go on scoring the ulcer. Like see 2, here it is for example 3, here it is 1. Okay, so on like 3, 4, 1, 3. Sorry, at one time at one scoring. Okay, so at the end we will get the average, and that average may be 4.2. Okay, so this is ulcer score, average ulcer score. Then we'll calculate ulcer index also. So this may be in terms of percentage, it may be 30%, it may be 20%, it may be 40%, it may be 50%, and at the end we can have average like 37%. Okay, similarly, percent inhibition also we can calculate here, and at the end we can take the average. So this average is of this control group, ulcer control group. Okay, then similarly we will calculate for normal control group, ulcer control group I have shown to you, and for standard control group. Then you will fill this. and then you can compare it. Okay? Compare with each other. So you will come to know that there is reduction in the treatment with the standard drug reduces the ulcerative index and increases the person's protection, ulcer protection. Okay, so this is what the experiment is. Now there is an assignment. Okay, and the assignment is what? You have to go through this paper. Okay, and later on we will discuss this paper. Okay, after some time, after half an hour, okay, we will discuss this paper. Okay, so I'll show you this paper. Yes, yeah. Is it visible? Yes, anyone?
Yes,
Yes,
sir.
sir.
Now see, this is a research paper which is written by Shahid Sahibu from Nigeria. Okay, so what he has done, he has used indomethacin-induced gastric ulceration animal model, and he has assessed the effect of spondius mombin and ficus expirata. Okay, these are the two herbal extract. So he has assessed these two extract in indomethacin animal model. Okay. So typically what you have to study is you just go through this abstract, try to understand it, right? Now just go through this introductory part. I'll highlight for you what you have to study. Yes, introduction, then just a second. Yes, introduction study. Just go through the introductory part, go through the abstract. Yes, go through the abstract and then go to this point directly. That is gastric ulceration, gastric ulcer induction. Then study this animal grouping. Then study this also. This. And this part. Okay, including this table also. Okay. Don't go into the details of this antioxidant study and all because this will be difficult. Then go through this table, this figure, this figure, this one, figure 2, figure 4, okay. And finally conclusion. Okay, so these are the important points and the points which are related to your experiment. So you just go through it. Okay. At the end, I'll give you 45 minutes. After 45 minutes, again we will come back and we will discuss these points. I am forwarding this on Google Classroom. Yes, anybody? Any doubt on this exercise?